Welcome everybody to our final Successful Ageing seminar for 2019. This year we've had 4 Successful Ageing seminars. I'm really delighted about that. It gives an opportunity to showcase the research that's going on at the University of South Australia. I'm Ruth Grant. I'm very privileged to chair the session, each session. And just the first thing to say is that if you have a hearing aid, could you set it to the T setting on your hearing aid, or else use the hearing loops that we have at the table at the front. Before we commence properly, I'd like to acknowledge that we're on the traditional land of the Kaurna people and that we recognise and respect their cultural heritage, their beliefs and their relationship with the land. A few housekeeping matters for those of you particularly who are coming for the first time, and if so, a very warm welcome. The first is to say that the toilets, always important as we get a bit older to know the location of toilets, are located on either side of the entrance at the bottom here. Males would turn to the left and females to the right to find the toilets. If you have a mobile phone, I'd be grateful if you would turn it to silent now. And also I'd like to stress feedback. We really do appreciate your feedback very, very much. There are feedback forms. You probably collected them as you came in. Please do fill them in. We do take them very seriously indeed. This is going to be a terrific session today. I know I've read something about both of the speakers and I think it's going to be absolutely fascinating. We have 2 highly respected researchers speaking today, Professor Emily Hilder and Associate Professor Albert Uhas. Just to give you a sense of the management of today's seminar, for those of you who are new, the first speaker will speak for 30 minutes. We'll have time for questions, then we'll have afternoon tea, return, and then the second speaker will speak for 30 minutes. Did I say 30 seconds before? Surely not. Surely not. 30 minutes. I wouldn't want to cut them off. And then we'll have questions and the session will finish around 4 o'clock. So our first speaker today is Professor Emily Hilder. Emily is the director of the Future Industries Institute. Now, if you've read the mission of that institute, and I think you have it with you, you will see that it's quite extraordinary in its breadth and depth. Create new industries through collaboration, transform the industries of today, and also to create the industries of the future. Now, you need a pretty impressive person to lead this institute, and that's exactly what we have in Professor Emily Hilder. She's an analytical chemist. She's Professor of Pharmaceutical Sciences at the University of South Australia. And she's also a member of the South Australian Premier's Science and Innovation Council. In 2014, Emily was included in the Analytical Scientists' Top 40 Under 40 Power List, and in 2017, one of the top 10 leaders in analytical science. Not only is she a Fellow of the Royal Australian Chemical Institute, but in 2018, and one of about 1 or 2 only analytical scientists, was made a Fellow of the Australian Academy of Technology and Engineering. Quite extraordinary, and perhaps it says something about her ability to research and to talk about that research across a range of different areas and have a significant impact in that regard. So please join me in welcoming Emily to speak to us today.
Thank you for that welcome, and it's a real pleasure to be here. I love the fact that people still talk about the top 40 under 40. I haven't been under 40 for a little while now, but it still makes me feel young every time it's mentioned, so it's fantastic. Today I'd like to talk to you a bit about what is the real passion that drives me in research in analytical separation science, which is fundamentally techniques that we can use to separate individual species in complex mixtures and to understand what is there and how much is there. So this is old technology, I would say, but how we use it and how we can use it in the future is where the excitement comes, because typically this is technology which is used in a laboratory with large instrumentation, large volumes of samples, very expert operators. We've taken some of this technology into the field where we can use it, where we take the sample, but the real exciting piece for me is how we take these measurement technologies to the individual. So we make this person-centric, we make it something where every user can use it. As I said to someone earlier today, we know what we're measuring what we want to measure, and we know that the data is good. Okay, so this is a really exciting challenge and one that's driven me for a long time now. And I want to talk to you about a couple of examples where it really sparked in me what you can do with separation science and why these technologies are important. One of them was quite deliberate. This one was the case of a really serious case of serendipity. I had a student come to me. He was doing a Masters of Pharmaceutical Science at the University of Tasmania, and he needed a project. And one of my colleagues said, "This hospital's got these samples of heparin." And I said, "What's heparin?" Chemist, not a pharmacist. And he said, "It's an anticoagulant, it's really important." And as you see here, it is one of the World Health Organisation's list. It's on the World Health Organisation model list of essential medicines. These are medicines that every hospital should have. And so it was really interesting. We did a nice piece of work. You'll see some of this work here, which we got a publication from. He finished his Masters. He went on to do a PhD. He's now an academic at the University of Tasmania. Great outcome for Rahul. And we did this nice piece of work where we took heparin and we treated it badly. We stressed it by putting it in acidic or basic conditions. And in this example here, we heat stressed it under quite extreme conditions. And when we did that, we could show that we took heparin that had this profile at the top, and we turned it into heparin with a profile at the bottom with, as we identified, an interesting peak that was unknown. There was an extra peak here. All very exciting for us. And then in 2007, it suddenly became really relevant. There was some deaths in North America that were attributed to contaminated heparin in hospitals. Over 200 people died before they recognised what was going on, and a guy in Canada Health found our method. He was reading the journal and said, "Hang on, that looks different than what we've seen before." He had the equipment to do the analysis, did this analysis, and within 24 hours they identified that it was a particular batch from a particular factory in China, and then later they identified what the contaminant was. But as it says there, they took the two batches off the market out of every hospital in North America, and there wasn't another death. And this was pretty, this was unknown to me at the time actually. And I found out when a former colleague of mine was sending me emails from a conference from the Canadian Chemical Society a few years later, when he said, "I'm sitting in this talk and this guy's talking about your work, and apparently it saves some lives." So I'm thinking, wow, that's pretty cool. Can you find out what really happened? So that's when we found out. It became a mandated method for the FDA for a number of years, but really taught me that sometimes you can make a difference without planning to. The next example I just want to mention before I move on to the main topic is one that was quite deliberate and close to home for me. I knew someone that was killed in this blast. This is the blast at the Kuta Club in Bali in 2002. October then, and this was a mixture of potassium chlorate, sulphur and aluminium. The blast was fortunately much less impactful than it should have been, and that's because the bomb makers weren't very good chemists, and they bought some potassium chlorate, and they also bought a large amount of potassium chloride, which was a beautiful inert filler for their bomb. But it was still incredibly destructive, as we all know. A large number of Australians died in this attack and a large number of people overall. And it fundamentally, I think, changed the way we think about security in the world. And This led the Australian Federal Police and others to come to us and say we really need technology that allows us to better look at these improvised explosives rather than military explosives. We happen to be experts in looking at inorganic ions, which are what these things are made of. So in the top left-hand corner here, we have that mix that was used, ammonium nitrate and fuel oil. This is your classic ampho mixture, as they call it, chlorate and sugar. You might see this as a demonstration with a gummy bear. That's fun. This is dangerous. And then some military explosives here as well. And we developed some new methods that could identify these markers. And in this case, was to be able to identify them at a site post the explosion, but led to some really exciting work where we said, well, wouldn't it be great if we could detect these before anyone has pushed a button? And that led, over almost 20 years now, to the development of this box here. It's called Grayscan. The first time I saw it in action, I was like a kid in a candy store. It was amazing, because what you have here is something that has separation technology that I never thought could be engineered into a box like this that can detect these improvised explosives, stuff like these swabbing systems at airports. And this has now been commercialised. There's been sales in 30 countries already, and we're excited to celebrate with a group of colleagues in winning a Eureka Prize for this technology earlier this year. So it's a very exciting one to go from what we thought was just some interesting ideas about separations to using them to solve a problem deliberately here. So with that context, today I want to talk about what I see is the vision for future analytical measurement, and specifically when we look at how we can bring measurement closer to the individual. Because this is a few years old now, this piece, but I really believe that future analytical measurement is going to be portable, it's going to be personal, it needs to be real time, and largely is going to be driven by interesting autonomous systems that may or may not be connected. And we've been really working for a number of years now to try and develop the tools that allow us to do this, and with a number of motivations in mind that I'll touch on as I move through this. And that's not to say that there's not things out there right now. In the top left-hand corner here is technology I worked on nearly 20 years ago in the US. I arrived in the US 3 weeks before 9/11 hit, and was working on a project on non-proliferation of chemical and biological warfare agents, which suddenly became very high priority after that attack. And one of the things that came out of that project was the microchem lab here, which was meant to be a handheld chemical analyzer. Other technologies that can be used for low-cost diagnostics to bring down the cost of healthcare. We have an example here in the top right-hand corner, and in the bottom here, which is some Australian technology doing something similar here, where you can actually detect this using a mobile phone. So really nice, simple technology. And in the bottom left-hand corner here, handheld technology for detecting, in this case, nerve agents. So there's a lot of really nice technology. But none of that yet takes you to the human, which is when we get into a little bit of a problem. I'm going to talk about this because this is some of the challenges I think have held back this technology. These are 3 pieces, all taken from the internet. I'm quite familiar with 2 of them, and one of them has just a nice story to it. On the left-hand side there is a beautiful design, a beautiful sensor that you can use to determine if your apple is organic or not. Now, I'm not sure how many people are going to be happy if they see you going around the supermarket sticking this probe into every apple. We bought this sensor. We found that it could not determine the difference between an organic and a non-organic apple. We pulled the sensor apart. It was attempting, I think, to determine nitrogen levels, but I would say that it is a complete fraud, but it's beautiful. But it is the kind of thing where they had really successful sales, and in fact they sold out to a much larger company. In the middle here we have a great news story from not that long ago, telling us about this fantastic 10-minute test for cancer. So this is from a couple of years ago. We can take it home with a drop of saliva. It's 100% accurate and so simple that you can carry it out at a pharmacist, the dentist, or in the privacy of someone's home. But strangely, we haven't seen this on the market everywhere yet. But this is the kind of thing that you see this and people go, oh, this is amazing. And yes, there is some interesting science behind it, but it's a long way away from a 10-minute test for cancer. But the one that really I'm sure a number of you might have seen about this, is the Theranos story. And this was a really interesting one where a few years ago, this woman, Elizabeth Holmes, dropped out of Stanford and started a company and got incredible investment in that company. And the premise was just absolutely fantastic. Whole drive. She had these, she said, we can take a drop of your blood and with our patented technology, we can test just about everything for you. And this is something that, you know, drives so many of us. For me, it's great. You know, I don't like needles. Most people don't like needles. There are some people where taking a large volume of blood is going to be problematic. This whole idea that we could take a drop of blood and be able to do this whole range of health tests. And in this case, you didn't even have to go to the doctors. You could go into a Walgreens drugstore, they would take your blood, and ultimately they were going to be able to analyse it there in these wellness centres. Unfortunately, while I think the idea is good, she was exposed as a fraud. It's still moving through the courts in the US. There's a fantastic book about it from a journalist who broke the story, a fantastic podcast called The Dropout, so I highly recommend, and even a documentary as well now. So unfortunately, it's certainly had an impact on it, but I put it up here because I think it's something, the way that people got around the Theranos story had so much to do with what was driving the public saying, we really want this kind of technology and we really want this to be true. So now I'm going to talk a bit about where this all fits in with my own research and what we've been doing for some time. And here I've just got some examples of the sort of materials we make. These are electron microscopy images of a whole range of materials, and really the message in this is that I like making porous materials out of polymers that we can use to improve analytical measurements. And the reason I'm showing you a range of materials here is the different structure of these materials allows us to analyse different types of samples. It allows us to separate different species based on their size, based on their chemical composition, based on their shape or other things as well. So we really are looking at how we can make different materials that allow us to have a whole range of tools to improve analytical measurements. We can make these in a whole lot of different ways. So we can make them with really very small pores and this very dense structure, up to things that have very large pores which we can use for separating and manipulating cells. So we can go from things that we could use for gas separations right through to things that we could use for separating cells and viruses and large particles. And by using a whole range of different chemical techniques, we can change the surface of these materials. We can put functionality into the material, or using heat or light, we can attach different functionality to the surface of that material. And this is really important in terms of controlling the chemical functionality. And I'll show you this as much as anything to say this is not a black box like Theranos was. One of the biggest challenges with Theranos is that nobody, even from the patents, could work out what they were doing. And I'm very proud of the fact that what we have is patented, it's open, and it's not a black box, and it can be challenged and tested by others. And we want to take these materials and put them in a whole range of different formats, because then we can use them for a whole range of different applications. So we can use them for things for standard chemical analysis here, in columns for large-scale separations, or in lab on a chip for the smaller scale. But what I'm going to talk about today is how we can use them for managing samples for analysis. So how we can use them for managing blood in small volumes, whether it's in a dried blood spot or, for example, in a standard type of PET format. And really what we're trying to do here is get to a point where, in this case, we move away from a large volume sampling to small volume, so like a finger stick, and then we're able to use this. So we use a material like those ones I showed before, which is what we have here in this example. This is not paper, it's a patented polymer material, which we can then use to handle very small volumes of blood in this case. It's an interesting story for me that I show you here a picture of a person, because when I started doing this work, it was actually motivated by the needs of the pharmaceutical industry, which was to start using less animals in the work that they were doing. And this was really exciting for me because I saw some fairly confronting things around the use of animals in that environment, but I was really heartened to see that we have an industry that is really keen to use as few animals as possible and treat them as humanely as possible. So all of this has come together in something which is just finishing up now, but I didn't want to mention it, which is the ARC Training Centre for Portable Analytical Separation Technologies. This has brought together this, and a lot of the things I'll tell you about fit under this. Training Centre refers to the fact that this is about training PhD students to become our workforce of the future. The best thing and the worst thing about Aztec is that all of the students and the postdoctoral researchers who have graduated from the centre have been employed almost immediately in industry. Twice I've had the, can you read my thesis quickly because I have to go and start my job. A number of them have been employed in the company, Trajan, that we started working with. Since I started working with Trajan, they've gone from 2 PhD trained scientists to 17. So I think this, and they're an wholly Australian-owned company, global footprint. I think this is a really exciting story about the value in training and taking this through to the products and to impact in the real world, as well as it's great to see students get a job. So I want to talk today about how we can go away from here, this traditional workflow. This is a typical traditional workflow with blood, where we collect a blood sample from a vein, we centrifuge it to separate the blood cells away from the plasma fraction, and then there'll be a whole range of steps that are taken to basically separate or manage components in that blood before we get to the analysis and data processing. And the timeline I've put up there for doing those steps is probably fairly generous. Often would take longer than that. These are slow processes simply because they take time, and the sample processing really is a limiting factor in a lot of these analytical steps. You'll see from that too, at every step of the way, there's an expert required. You need a phlebotomist to collect the blood. You need someone who can process the blood. You need someone who can do the final analysis. And we're trying to walk away from that, that kind of workflow and move to a workflow where it's less invasive. It puts it in the hands much more of the individual and allows us to take this kind of testing to people who aren't necessarily living in major metropolitan areas. So now, instead of having to have someone to collect the blood, we can do this with a simple finger stick. We can manage a whole range of these different processes through fluid handling, and I'll talk about that. And then we should be able to take it again to the final analysis. This last step is the one where you would still have an expert operator, but that's simply in terms of managing the quality of the data, being able to get such high quality analysis. I want to talk a bit about some of the steps in this, and the first one here I'll talk about is one that we've worked on to be able to separate blood cells from blood without needing a centrifuge, because it's really hard to miniaturise a centrifuge. Big and heavy. So instead what we've done is we've made a material that when we aspirate the blood through it, it's a very simple low pressure pipette, you'll see that the blood cells stay immobilised on the material, which is a function of chemistry and the size, so that porous structure of material, and what we get at the top there is this really nice fraction plasma. So now we have something where we can take even 20 microlitres of blood and we can separate the cells of the plasma. This is really nice. Here we can do that with a normal blood cell, where we see the normal red blood cells, we can see the white blood cells, so we can get some fractionation. What's even nicer, and I haven't got any results that are good visually here on this, but what's even nicer is we've learnt that we can use this to look at blood cells that are in some way impacted, and in particular in terms of malaria. So we can stage a malarial infection to some extent by how we can separate these cells. When the malaria parasite infects a red blood cell, it changes the flexibility of that cell. And as it goes through various stages, the blood cell becomes more and more stiff, and then it can't move as well through these porous structures. So we can take this and identify whether blood cells are at an early stage of infection or a later stage of infection, and in that way help to understand more about this disease and help hopefully to develop a diagnostic. So there are multiple options for this sort of technology. As I said here, we can do this really nice blood fractionation. We can see the white blood cells here at the front of the material. We can find the red blood cells here when we fix them and look at them under the electron microscope. We can release them off the material and collect them. So it's a really nice way of being able to do that simple step of fractionating the blood cells from the rest of the blood. And we've done this because we do a lot of work where it's really important to be able to analyse things in plasma. Here's just a simple example. I'm with one of my former PhD students, Esme, who is now working in one of the major analytical instrument companies in the US, where we developed a material where we started with this material here, which you can see is very hydrophobic. It's a water droplet sitting on there, which doesn't penetrate at all. We coated it with a hydrophilic coating, and what that does is it makes it protein repellent. So now we have a material where, in this example here, the much larger signal, that's using the protein repelling material, where we can now search for a drug in the plasma, which is a very high protein concentration, and get a nice accurate measurement of that drug in plasma. This is a nice simple example here of ibuprofen and being able to determine that very, very simply by tuning the material we use for that analysis. Okay, with a small drop of blood. But a real motivation in working with small blood volumes has been able to do more with dried blood samples, because there's a lot of reasons why it's nice to work with dried blood rather than wet blood. And one of those being that there's been a lot of work done since the 1960s in using this technology, in particular for neonatal screening. So every baby that's born in Australia has this done, where their blood is collected from a heel prick. It's spotted onto a filter paper, and it's analysed in particular to determine things like PKU and other errors, inborn errors of metabolism. So fantastic technique, but in this case it's a yes or no. So it's not about a particular level and a quantitative result, it's are the markers present or not. So we started working with some new materials, as I said, with these polymer materials, so we could try and take this technology to being something that could be used for quantitative analysis. Paper's great for qualitative analysis, but not so great for quantitative analysis. Paper's great because you can take it and spot it on a card, and it's very easy to use. You can punch out a spot for your analysis. So the format's great. And of course, in the other examples you saw, we've been making these materials not in flat sheets, but in these other more analytical formats. So it was a bit of a shift for us to start making this material to look like paper. It started with a conversation with some scientists we were working with at Pfizer, where we said, well, what if we made it on a piece of backing, and then it might behave like paper? And lo and behold, it did, which was great. And another nice thing we found is that because we were making this very well-defined synthetic material, it behaved even better than paper in some other ways. And one of the ones I just want to point out here is over in the bottom right-hand corner, is one of the big challenges in blood analysis is that not everyone's blood's the same. So if you've got over 50% red blood cells, you will not be allowed to ride in the Tour de France, just so you're aware, because it's too high a concentration to be safe, and a suggestion that you may be blood doping. If you're very young or very old, you may have a high hematocrit, or if you have particular health conditions, or you may have particularly low hematocrit, concentration of red blood cells. And so we have to be able, and that fundamentally changes the viscosity of your blood and then how it behaves as a fluid. What we were able to do here was show that when you go from 20% all the way through to 80% hematocrit here, so representing that significant increase in viscosity, on this material we're able to have a spot which is essentially the same size, which may not seem important until you realise that the standard way of analysing these is to punch out a section of particular size. And if your volume is changing of your overall spot, then you can't get the accuracy when you punch out a spot. So this was a real game changer for us in being able to use these materials. I'm going to show you some examples for how we use them, because that's been an exciting part of it here. First one here is around fatty acid profiling for health. This is something that's a really good health indicator and has also been demonstrated by researchers here locally at SAMRI that is a great indicator for pregnant women in terms of their chance of miscarriage. And so you can see that fortunately we're not in the red yet. Red is not good in this case, but certainly we're not in the green in terms of the known fatty acid profiles within our health. And this relates in part to hereditary factors, but also very much to diet. So there's some materials out there where you can buy them and use them for measuring this in the home. Unfortunately, this is not showing you measuring these fatty acids. What it shows is levels of contamination on the materials you use to do this analysis. So you probably guessed big is not good. And if we take all of the materials except for the one on the far left-hand side, these are all commercial materials, including the best in class, which shows you that no matter how well you do the sample collection and the analysis, you're already in trouble because there's huge contamination, and thus you might be told that you have a particularly poor result when actually there isn't any issue at all. And so what we discovered with our materials is we had these very nice clean materials with low levels of contamination. And the reason I'm showing this is because one of the things that we need if we're going to have good tools to allow us to use these for genuine health monitoring is to know that what we measure is really what we measure, and that it's something that people can use without sending someone a home kit with a protocol saying wash 17 times under pure distilled water and then dry in a vacuum oven, which is not going to suit most people in their home. So we've done a bit of work here again on showing that we can take these materials and again and again and again, got them here on the right-hand side. They're cleaner, they're easier to handle, and we don't have these same levels of contamination. It's probably not that surprising that when we look at fatty acids, paper is more contaminated than a synthetic material, because paper, of course, is a natural product made from cellulose. We can use this then to be able to look at the extraction efficiency and stability over a range of time, and with that get a very good material for this health monitoring. And this material is now being commercialised for just this application. Another nice thing about this material is while we can use it in one case for looking at these small molecules like fatty acids, we can also use the other component, which is the red blood cells, to look at things like, in this case, glycated haemoglobin. Glycated haemoglobin is a fantastic marker because it's the best medium to long-term health marker for diabetes. And it used to be that if you wanted to test someone for diabetes, you challenge them with glucose and measure their blood sugar. Now you've got two choices. Your first one would be to test the HbA1c and then to do a glucose challenge test, because those two bits of information together tell you about the last 3 months in terms of their health, as well as the immediate impact of a glucose challenge. So very, very important when we want to identify and be able to help people live with these metabolic diseases. So in this case, we can take the red blood cells that we've separated from the plasma, wash them through with an appropriate buffer, or even just water in this case, and we're actually able to use this collected blood to be able to measure HbA1c accurately. We can compare this to whole blood. We can see that we have a really good fit here, and that we are able to correlate these results really well. And this is nice because you could envision collecting a blood spot and using it for more than one analysis. I didn't say before, one of the really nice things about dried blood is you can put it in the mail. As soon as it's dried, it's no longer harmful. It doesn't need to be kept cold. You don't have to worry about pathogens and viruses. It immediately becomes a safe sample. So this becomes a real game changer in being able to collect and move samples around. Well, the last thing I just want to talk about is that it all looks really simple until you start thinking about the practical challenges. And the reason I'm pointing this out again is because whilst I'm disappointed in the damage that something like the Theranos story has done to the opportunities in this space, I think it's just shown us that we need to just consider all the factors when we're developing these new technologies, because we know that the premise of being able to measure things with small volumes of blood is a really good one, and one that is going to bring health benefits to many, many people. We'll see much more of that in what Albert's going to speak about after the break. But in this case, we did what someone thought was a very boring study, turned out to be very important, showing how we could collect an accurate volume of a really small amount of blood. You're trying to collect 3 microlitres of blood. And what we learned, particularly from colleagues working in the pharmaceutical industry, is that the biggest problem they had with introducing these small volume techniques was that, as one of them put them, no one knows how to use a pipette properly. All the variation was in who used the pipette. And so we developed, and I'll show you a bit more about it here, we developed a device that allowed us to collect an accurate volume of blood, which then allows us to take this factor out of it. And we tested this. We tested it against the standard pipette, the one that nobody knows how to use. We tested it against a positive displacement pipette that was automated, so it should be the same for everybody. And we tested it against our preferred solution in our device here, which is to fill a capillary end to end and then transfer the volume of that to a piece of material, so a piece of paper in this case. And we did a study where we compared each of these 3 ways. So we compared each of this, the pipette, the digital syringe and the capillary, and we compared it at different, across different blood samples with different amounts of hematocrit. And we spiked it with caffeine, one of our favourite analytes, and then we tested to see the difference. And I just want to show this because this is not an example of how terrible different people's pipetting skills are, but an example of how we can use technology to take that human error out of the equation. When we compared across 2 operators who were working with a set of instructions rather than knowing a special trick way that you should use it, so there's working with a set of instructions as someone would be if they were blinded to it otherwise. We showed that we had this incredible variation between the 2 operators who would both claim that they're experts in using pipettes. When they used the positive displacement pipette, this digital syringe, we got a much better fit. And when they used the capillary, we got this very good agreement. And we've now seen this across many more users as well. So this was something that seems like a very small thing, but is really important to how we can develop technologies which we know are robust and can give us the right analysis. And this is the cornerstone of here, HemaPen, which has been used in a number of trials and in a number of research settings, which is the device that came out of a lot of this work. It's been developed and commercialised by our industry partner, Trajan Scientific and Medical. It's a company that is absolutely driven by bringing new technology that can benefit people. It really is fantastic to work for, and it's fantastic to see that that's at the core of what the company is doing all the time. And what's nice here is that we've got a very simple device modelled on the big 4-colour pen, which at the time the postdoc who came up with the idea was sitting there at his desk clicking the pen, thought, maybe we could use this to collect blood. So we have a device with 4 capillaries, 4 collection points that each can collect 3 microlitres of blood, and we can collect this simply by touching it to a drop of blood on a finger, because the capillaries draw off the blood and transfer it completely to those materials. What's nice is after you've collected the blood, you click it back into the holder, and it can be sent away for analysis. And we're using this now at Women's and Children's Hospital in a paediatric study where we have some very excited parents who are looking forward to the day when their children can have their blood samples taken just with a drop of blood. And just to show you, I've said that one of the most important things for all of these technologies is it's tested by people who didn't invent it and can't influence it. So this is the paper I'm most proud of that I had nothing to do with. This was done by a group in Belgium who I don't know at all, but I deeply respect their work. And they've been able to demonstrate in this work that if we use a standard blood sample, we get These are the red dots that are spread all over the place. So if we take a blood sample and we make blood spots on a standard card, the red dots represent the spread of results. And if we use the HemaPen, where we can accurately control the volume, the green dots represent spread of results. The line at the top shows you the variance in the green, the line at the bottom shows you the variance in the red, and you can see that we really do have a technology here that allows us to have much more accurate analysis and know that the variation we see in our analysis is due to real variation and not due to our experimental design. So very, very exciting and very pleasing to see this work. So that's all that's left now is for me to thank many people who've been involved in this work. This work started when I was at the University of Tasmania. I moved to University of South Australia 4 years ago, and it's been a fantastic move for me. We've had support from many facilities and industry partners, but the real people to thank here are the people who have done this work. Ricardo here, who has now started a new job in Germany. He's from Portugal, came here to do his PhD and has now moved back to Europe. Sarah, who had a little break from her PhD but has a beautiful daughter to show for it, and has been doing some really fantastic work which is strongly aligned to what Albert's going to talk about. James, just finishing his PhD now. He joined just after a long career as a medical scientist in Malaysia, and has been working on the material for blood separation and staging malaria. Neeraj here, who has been working on a number of these technologies as well, he's just finished his PhD and is spending some valuable time with his grandparents in India before he comes back to start a new job with Trajan. So it's been a fantastic journey. This is the team at the Future Industries Institute, and thank you all for your attention.
Emily, thank you very much. That was absolutely fascinating. We have time for questions, so we have a roving mic. I know Emily would be delighted to answer them, so fire away.
It's a really good question. So I'll be really honest with you, I'm not at all driven by the financial reward. No, I'm not, because I love my job. I love the fact that I get to come to work every day and do something that's fun. I feel kind of privileged, actually. But absolutely, most universities will ensure that they recover costs on the cost of commercialising something. That's a really interesting example, because that was licensed very early to a commercial partner who took all of the risk. Grey Innovation, who have taken that through and formed Greyscan, I wouldn't even want to think about how many millions they've spent taking that to market. They've taken incredible risk. It's a fantastic Australian success story. But one of the things that's been really exciting is that it has supported almost 20 years of research across a whole range of areas, with that and related areas. So some of the reward is in that future research piece. I've been working with Trajan now for about 10 years, and we continue to work together. So that's a reward in itself when we work together on further research and they help to fund that. Ultimately, in a number of technologies, you'll see that commercial income flow through, but there's probably few and far between, because the cost of taking something from a great idea in a lab through to a product is not cheap, but it's great when it does happen, because the impact is fantastic. I'm pretty excited too when it turns into jobs for people.
Maybe we should repeat the question.
So the question was, has the work spread into the area of picking up drug cheats in international events? We haven't deliberately worked in that space. We did do a little bit of work at one point around horse racing, because we're very good at cheating in horse racing in Australia, apparently the world's best at cheating. As it was put to me by someone from a Singapore testing lab one day. The only reason we haven't gone into that space has been very much because of the industry partner we work with, who would say to some extent, whilst that's a very interesting space, it's not directed towards helping deliver good benefits for people. So we've focused on applications where we felt we could have a greater impact in improving health for people rather than catching people who are cheating in sport. That doesn't mean you couldn't use the same technology. I guess it's about where we prioritise our effort.
This is slightly connected to drug cheats. You talked about the Tour de France and when people get over the magical number of 50. And now Pantani was in the Giro, which is the Italian equivalent, and he was caught, of course. And he
was close to 60, I think, when he got caught. Yeah, a magic of 60. Yeah, it's in all UCI events, and it is because it puts an incredible load on your heart, and I suspect that was probably part of the problem with Pantani. He had pretty thick blood.
How is it then that we can get the good ones, such as you people, to be able to get ahead of the medicos or the medical advisors for sports people in, as we hear with cheating in cycling, for example?
That's a great question. I think the key here is when you start seeing things like the athlete's passport. So when you start having routine monitoring of people where you have regular testing, and that's where some of these technologies are really important, because if you can take a finger stick sample and you can do it every week or every few days and demand that someone has that regular testing, it protects the honest athlete, because you will see an anomaly quickly. You're not then trying to... We're all individuals, so you're not trying to say, oh, well, why is your level this? And you say, well, it's always like that. You know, talking to someone recently who said, oh, I thought I had a high cholesterol reading and then I realised it's been the same for 8 years when I talked to my doctor. So it's not really high, that's just me. And I think it's the same with athletes. When you're doing a single test and you're saying, oh, your level is high or low depending based on an average population, it doesn't compare to others. It doesn't make it fair. And so then you're then looking for differences. And if you can do that where you're looking for differences, you've got a better chance of catching people beforehand, because you're looking at the impact on their physiology rather than looking for the drug when you haven't even realised they've started using that drug yet. So I think that being able to do regular and testing like that, honest athletes will appreciate it, and they already talk about it as being something great because they've got nothing to hide, and it makes it really hard for a cheat.
One last question.
I have a little self-test INR CoaguCheck unit, which I use regularly and which is very useful if I happen to go away on holiday somewhere where I can't get INR tests done. Is your work leading towards making more of these for different areas of blood testing?
Absolutely, that's what we would like to see. There's that kind of example. The other one that we've worked with a little with the Royal District Nursing Service has been around being able to provide better health support to homeless people or people in more difficult populations to access. If you have something, all the evidence shows if you have to do a test, take it back to take it to a doctor, wait for a result, come back, you miss a certain amount of the population. So we'd like to see that for a whole range of different tests. We're going to have to put some pressure on the healthcare model to do that, but I need another hour to talk about that. Because look, the Medicare model in Australia, I'm not saying it's right or wrong, but like many countries, is set up around tests being sent to central laboratories, so it does require something of a reset of the healthcare system to incentivise having these tests available remotely.
I think we might have to close it off there, but I know that Emily is quite happy to answer questions during the afternoon tea break. I trust I haven't assumed that. And so there will be the opportunity for those who wanted to ask questions now. It's my pleasure to introduce now Associate Professor Albert Uhas, who holds the Barbara Hardy Chair in Environmental Science and Engineering. And he's also a strand leader or theme leader in the Future Industries Institute. If I put it simply, Albert Juhasz is a world leader in the assessment of human exposure to soil contamination. Let me elaborate just a little bit there. He and his team have developed the world's most accurate way to gauge exposure to soil contamination after the land has been used for mining, industry, agriculture, and waste management. To date, for safety purposes, every contaminated site has been assumed to be 100% likely to cause harm, thereby requiring remediation to the fullest extent, with the spend estimate in Australia alone annually the order of $2 billion. And this is because there hasn't been a reliable way to measure the contamination risk to humans through soil contact. And that contamination risk to humans through soil contact is known as contaminant bioavailability. Albert and his team have developed assessment tools that measure the contaminant concentration and predict which levels will be harmful to humans. Hardly surprising, then, that environmental agencies, including the US Environmental Protection Authority, have been particularly keen to learn from what Albert has developed with his team, developing a more sensitive gauge for managing and remediating soil, thereby delivering significant savings worldwide. Please welcome Associate Professor Albert Juhasz as he speaks on Eat My Dust. Thank you.
Thank you very much, Ruth, for the introduction. Now, just checking at the back, can we hear okay? Is that a thumbs up? Excellent, fantastic. So thanks, Emily, for setting the scene in terms of how we can potentially assess people's exposure to contaminants through microsampling of blood. What I'd like to do is take a step back from that and say, well, how can we predict people's exposure to environmental contaminants by having a look at the contaminant source, the exposure source, whether it's soil, dust, food, water, before people actually get exposed. So just a few points about myself as a way of introduction. I gained a PhD in environmental microbiology at Victoria University of Technology in Melbourne and have worked at a number of different organisations, both in Australia and overseas, in the environmental microbiology space and also in environmental contaminants. I've been at UniSA for almost 17 years, coming up 17 years in February, with my main research focus looking at environmental contaminants and remediation technologies, and more so over the last 10 or so years to look at human health exposure to these contaminants and how we can predict that. So when we consider environmental contaminants, what are the first things that crop into our minds? Maybe it's industrial contamination. This is a historic photo of Port Pirie showing atmospheric pollution. Some may say it hasn't changed so much over the last 125 years. This one is the Exxon Valdez accident in Prince William Sound in 1989. That was, in fact, the point in history that got me interested in environmental science and bioremediation. And the third example up there is potential mine site impacts, whether it's soil contamination, water contamination, and so on. So these are the things that often jump into our mind when we think about environmental contaminants. But there's other things. We often don't think about indoor environments as a source of contaminants, but, for example, The chairs that we're sitting on at the moment, they may be your couches in your homes, often have flame retardants or retardants put onto them to protect them from stains, etc. So there's a number of different brominated and fluorinated flame retardants that may cause some issues if you're exposed to elevated concentrations of these. And over the years, some of these have been banned. There's potential contamination in water, not just from water sources, for example, in Bangladesh and West Bengal, India, that are contaminated with elevated concentrations of arsenic from natural sources. The contamination in the water may come from the plastics. It may come from materials that are used in the plastic production, such as antimony. There's insecticides, herbicides. There's been a lot of work and a lot of media attention over the last 12 months about glyphosate and the potential adverse health effects associated with exposure to that. There's products that we use in our home to kill flies, insects, and so on. There may be homegrown produce or produce that you get from the markets or supermarkets that may contain herbicides, pesticides, etc. And also there's personal care products where there are a range of different organic constituents in these that may have an adverse health effect. So the way that we can be exposed to these contaminants is through a number of different exposure pathways. So you may be inhaling volatile materials. It could be through the inhalation of dust, dermal absorption through the skin, or maybe a known ingestion, so you might be consuming food and water, or it could be incidental soil ingestion, as in the case of kids where they're crawling around on the ground and then dust accumulates and sticks onto their hand and then it's transferred into the mouth. And we don't have to go far to see multiple examples of these in the newspapers. So I would say almost every day there is something in the paper about environmental contaminants. And I've got a couple of examples here. So if we look at the New York Times, earlier this month, New Delhi choking on toxic air declares health emergency. And if we go back 2 weeks ago, there was something in the paper to say that Sydney had the worst air quality in the world arising from the bushfires. In the UN news, soil pollution jeopardises life on Earth. There's issues associated with contaminant uptake into plants. There was a recent survey done in China that showed that there's approximately 15% of arable land has got elevated concentrations of contaminants in there above guideline values. A little close to home, a landmark class action over PFAS contamination in Australia, announced by Erin Brockovich, who is famous for the work that she did in terms of a class action in the US over a chromium-contaminated water. I'll be speaking a little more about per and polyfluoroalkyl substances. It's a little bit of a mouthful, and the issues that we do see in the analysis of those compounds. And the last one from CNN Health, 95% of tested baby foods in the US contain toxic metals. So these issues aren't far from home. So there are a wide variety of different exposure sources. We can be exposed to these contaminants from a number of different exposure pathways. I'll be talking a lot about the incidental ingestion pathway, which is particularly pertinent for children who have got the activity and hand-to-mouth transfer. But if we go back in history, Paracelsus has made the quote, "The dose makes the poison." We need to understand the dose that an individual is receiving to understand the risk associated with that exposure and what the potential health effects may be. Now, understanding exposure is extremely complicated. There are a wide variety of factors that can influence how someone is exposed to a contaminant and how much of that may be absorbed into the bloodstream. So something that isn't on this slide is the socioeconomic status of the individual. So this will drive where the individual lives, the type of house, if there's leaded paint in there, is there asbestos, if the house is leaky with dust infiltrating in, whether they live close to a main road where there might be exhaust fumes, whether they live downwind from industrial facilities and so on. But other things that can influence exposure include behavioural aspects. So again, if we're talking about a child, how they interact with the surrounding environment, whether that's inside the house or outside the house. The personal hygiene of the individual, how clean the house is. So this will all drive exposure to potential contaminants of concern. There's the physiological and nutritional aspects as well. So whether you haven't eaten all day, you're exposed to an inorganic contaminant, that means you've got a low pH in your stomach because you're under fasted conditions, which may then facilitate the solubilisation of that contaminant and potential absorption into the bloodstream. Nutrients, nutrition, the type of food that you eat, there are a whole range of different essential elements that can compete for uptake with the non-essential elements, the ones that we don't want, such as arsenic, cadmium and lead. So nutrition can play a significant role in terms of contaminant uptake. And then there's the physicochemical parameters. These are the ones that someone like myself, as an environmental scientist, we find these easy to measure. So we can assess the contaminant in the contaminated matrix. We can look at the mineralogy, the speciation of the element that may be in that matrix, the surface area, how big the particles are they're exposed to, which will influence solubilisation, the impact of pH on the solubilisation, and then the interaction between different elements or different constituents within food and the contaminated matrix that may influence the uptake of the contaminant. So what I'm talking about with exposure is how much of the contaminant you're exposed to, you're physically in contact with, and then how much of that, once it's been internalised, can be absorbed into the bloodstream. So this is a diagram looking at bioavailability. So it describes that if you're exposed to a contaminant, whether it's a soil-borne contaminant or it's in food, once it's been ingested and it goes into the stomach, there's a number of processes that will lead to the release of that contaminant from the food matrix, the soil or the dust matrix. And depending on if you're talking about organic contaminants or inorganic contaminants, then that may be facilitated through either the conditions in the stomach or the conditions in the small intestines. But needless to say, the contaminant needs to be solubilised for it to go across the intestinal epithelium to be absorbed into the systemic circulation. Once it's been absorbed, then it may accumulate in different organs, whether it's the liver, the kidney, adipose tissue. It may have a long residence time in the bloodstream, it may partition into bone. And then depending on the contaminant that you're exposed to, depending on the type of exposure, whether it's acute and chronic, where it accumulates, there are a whole range of different health effects that may ensue. So all of this is termed bioavailability processes, the processes that lead to the absorption of the contaminant into the systemic circulation, where it can then cause and affect potentially an adverse health outcome on the individual. So the question is, why is bioavailability important? Well, if you're undertaking a risk assessment or an exposure assessment at a contaminated site, we have this formula, so you're looking at chemical daily intake. There's a whole range of parameters that you include in there, including the concentration of the contaminant, for example, in the soil, the ingestion rate, body weight of the individual, exposure time, and so on. So what this actually does, it only tells you about the external dose. It only tells you how much you were exposed to and not how much is actually internalised once it's been ingested. So what we need to do is determine the relative bioavailability. So if we include in relative bioavailability, we know what the effective dose is. So if we go back to our Greek philosopher, I think we need to change his saying and say it's the effective dose that makes the poison. So how much is internalised that then can have an effect on the individual? Now, we're talking about human exposure to environmental contaminants. Can you put your hand up if I said, look, can I run a quick experiment on everyone in here? We want to look at arsenic bioavailability in a contaminated soil. Who would ingest some contaminated soil for me? No, there's no one. Okay. I would, because I know the risk associated with that. So you think human health exposure assessment, the most appropriate receptor to test this is in humans. And this work has been done. There's been some work done probably the last few times in the 80s and the 90s, where things like exposure to arsenic or exposure to lead, you can do relatively simply through semi-non-invasive methodologies. So this links into the work that Emily's been talking about in terms of blood analysis. You can look at concentration of contaminants in blood. You can look at urinary excretion. So we can do this for things like arsenic and lead. The problem is if we're talking about other contaminants, whether it's cadmium, or you're talking about fluorinated flame retardants or brominated flame retardants or things like that, you may have to look in these other organs. So you may have to look in the kidneys and the liver or the adipose tissue or in bone. So unfortunately, no one volunteered here for the arsenic test. I'm sure no one's going to volunteer to have a kidney biopsy or a liver biopsy. I certainly wouldn't. Of course, there's ethical issues associated with testing in humans. And so as a consequence of that, a number of different animal models have been utilised for the assessment of contaminant bioavailability. Again, there's challenges associated with doing in vivo research, ethical considerations. But a number of different animal models have been utilised. For example, pigs have got a physiology that's similar to young children, some children more than others. The bone weight to body ratio is very similar. They're monogastric omnivores. They've got a large blood volume. You can take repeated blood samples and do the traditional methods of analysis as Emily first outlined at the start of a presentation. Of course, it's very expensive to do that type of study. So other methodologies have utilised mice. So you've got small blood volume in a mouse. Again, issues that Emily outlined in terms of small blood volume for analyses. You can't do repeated blood samples, or it's difficult to do repeated blood samples in the mouse, so you use larger animal numbers. As I've mentioned, there are issues associated with this, so there's the ethical considerations. So I want to refine our methodologies. We want to replace the methodologies and replace these for other non-animal based approaches. They are very expensive. So, for example, if we were to undertake a pig study having a suitable number of replicates, for a contaminated soil, if we're thinking about something relatively simple such as arsenic, it may be in the order of $20,000 per soil. So it is quite expensive. There's complexity associated with these assays. There are standard operating procedures for individual organisations. However, there isn't a standardisation of methodologies across different organisations. So, for example, the way that we've run in vivo studies for swine is that we'll gavage the animal and it will be a single dose exposure, monitoring blood arsenic concentrations for a 26-hour period, whereas other studies in the world, they've dosed the animal repeatedly for a number of days and then looked at urinary excretion. So there's differences in terms of the potential outcomes associated with these methodologies. So I'd like to present two different examples of how we can utilise bioavailability and in vivo studies for the assessment of both legacy contamination issues and also issues associated with emerging contaminants. So this is some work that we've done at the University of South Australia looking at arsenic bioavailability in two different locations. The first one is in the central goldfields of Victoria. So if you're looking at ore deposits or gold deposits, they're associated with sulfidic material. arsenic is commonly associated with the gold. And so when you're releasing the gold for recovery, you also release arsenic into the environment. The second one, this picture here, is sugarcane plantations in Hawaii. So we were approached by the Hawaiian Department of Health. They utilise arsenic-based herbicides. Historically in these regions, arsenic has built up in the soil. They're no longer undertaking the sugarcane growth of plantations in that area, and they're redeveloping the area for housing. So the concentration of arsenic in those soils were elevated, higher than a guideline value. They wanted to assess the bioavailability to see whether or not the risk is lower than what is actually perceived. So the approach that we utilised swine as the animal model. We monitored the blood arsenic concentrations after exposing the animal to the contaminated soil over a time period. And this figure here just shows that once we've had administration of the soil into the animal, the contaminated soil, we can monitor the blood arsenic concentration over time and calculate what the area under the curve is. And then we can compare that to a known reference dose. So in this case, it's sodium arsenate, which is what the toxicity reference value is derived from. And we can compare the area under the curve from here to that and dose normalise it. So what we found in the case of our Victorian soils, which is the soil number 1, the mine impacted soils, and the Hawaiian soils, which is soil number 5, is that the bioavailability in those soils was low. They're somewhere in the order of 10 to 15%. So if you were doing a standard risk assessment at these contaminated sites, the assumption would be that all of the arsenic would be bioavailable. So in this case, you're an order of magnitude less in terms of potential exposure. So this has a huge impact in terms of how you're going to treat the land to get it to a point where residential development can be undertaken. So if you're assuming that the contaminant was 100% bioavailable, you might be forced to instigate some remediation technologies to minimise that risk. Here, we're first understanding what the exposure is that drives risk, which then can lead to whether these remediation or risk management strategies need to be implemented. Now, we can understand what's going on by doing some supplementary investigations to understand the factors that are driving the low bioavailability. So down here, we've got some analysis that was undertaken on those soils to look at what form of arsenic was there. Now, it's probably very difficult to read those different names of the different arsenic minerals from the diagram, but needless to say, the low bioavailability in the mine impacted soil was because the arsenic was associated with the sulfidic material. So inherently low solubility. So when it's internalised and is sitting in the stomach of the pig, even though it's a low pH condition that can stimulate solubilisation and dissolution, because those minerals have got extremely low solubility, that's why the bioavailability is so low. In the case of our herbicide-impacted soils from Hawaii, it was the iron content that was driving the low solubility. All the arsenic was sorbed onto iron oxides. Now, Hawaii is well known for volcanic soils, very high concentrations of iron in these soils, but it was the form of iron that was in those soils and the way that the arsenic was attached that meant that there was low solubility in the stomach and low bioavailability. Now, the second issue I'd like to talk about for the in vivo studies is the issue with the per- and polyfluoroalkyl substances, or PFAS. Now, again, if you're into the papers over the last couple of months, you may have seen some articles talking about the class action that's happening in Australia, where a number of people are suing the Department of Defence and the federal government regarding the use of the firefighting foams to control Class B fires or fuel fires. And these compounds getting into waterways and impacting drinking water, impacting the quality of food, and potentially having health effects on individuals. There's been a lot of hype about the PFAS in the firefighting foams, but PFAS are also found in a whole wide variety of other consumer products. So classically, you'd think of non-stick frying pans where these fluorinated compounds are utilised so that your eggs don't stick and your bacon doesn't stick for your morning breakfast. You've got in paint products, in shampoos and so on. Now, the chemistry associated with these compounds is extremely complex. This is one compound that I've got up here. It is called PFOA, the acronym for it. You've got a number of carbon chain length. It's fully fluorinated with the green fluorines indicated here. And then you've got a functional group on the end, which is a carboxylic acid. Now, that is just one of many classes of PFAS that vary in carbon chain length, they vary in functional group. There might be ethanols, there could be acetic acids, sulfonic acids, and so on. And there are over 4,000 of these compounds. So analytically, it's very challenging. We monitor for 28 at the moment. So I stole this concept from a presentation that I saw just a couple of months ago. Good old Donald Rumsfeld had the famous quote about known knowns, unknown knowns, and unknown unknowns. And we can relate this to the issue with PFAS. So our known knowns with the PFAS are we monitor for 28 compounds out of the 4,000. So we know what they are. We can quantify the concentration of these in the matrices that we're looking at. There are known unknowns. We know that there are precursor compounds in a contaminated material that can be oxidised, whether it's by an individual or an animal or in the environment, to our known 28 PFAS compounds. But then there's also the unknown unknowns. So there's unknown compounds that we're not quite sure of that could exert a toxicological response, not that we know what that is. And also, there could be precursor compounds in there that transform to these unknown PFAS. So it's an extremely complex system and chemistry that we're dealing with, but it's an issue that is pressing at the moment, particularly for Department of Defence. We've undertaken some of this work within the Fitch Industries Institute just to get an idea about if an animal model is exposed to these compounds, where it actually goes. So here we have examples of a 4-carbon chain length sulfonic acid, a 6-carbon chain length one, and an 8-carbon chain length one. So the animals were administered these compounds, that was it, and then we monitored where it went. So if you're looking at the 4-carbon fluorinated compound, it is absorbed and then it gets excreted in the urine relatively quickly, so the metabolism of that compound is quite rapid. If we just add 2 carbon chain lengths onto the end of that compound with the fluorine, then it doesn't really come out in the urine anymore. It accumulates in the liver and it also accumulates in the muscle. So these compounds like to be associated with protein-rich material. But again, when we increase the carbon chain length up to 8, the distribution changes. So more we want to go into the liver compared to that of the body. So we need to have an understanding of the pharmacokinetics of these compounds before we can undertake develop methodologies for assessing their bioavailability. So we've done this with a number of different contaminated soils, which are indicated up here, and this is for PFOS, so the 8-carbon chain compound with the sulfonic acid on the end. And we can see, depending on the soil that we run, it's the physicochemical properties of the soil that can influence the bioavailability. Now, the good news is we can add things into the soil that can reduce the bioavailability. So this was work that we did for a local SME called Ziltech, where they have remediating products that you can put into the soil, bind it up, reduce leachability and reduce the amount that is absorbed into the bloodstream. Now, when we look at the 8-carbon chain length compound, which has a carboxylic acid at the end, we find that we've got this. We've got our known unknowns happening because there's something else in that soil that is being metabolised, that transforms into that compound because we're getting recoveries of the PFOA in the order of 130, 140%. So we don't know what it is, but the animal is making it in its body. And again, that could have greater impact if you're just analysing for that single compound in your environmental matrix. Again, the good news is we can add our remediating products into the material, into the soil, and you get a reduction in bioavailability. We've done a lot of this type of research for a number of different contaminants, a number of different matrices, whether it's soil, contaminated food, water, and also lipstick. So this was an interesting study done with collaborators in China where they were looking at lead in lipstick products. Sorry, ladies, but potentially you can consume up to about 5 kilos of lipstick in your lifetime. And so we were looking at what might be the lifetime exposure to lead in lipstick products. A little hint, buy the more expensive ones, not the cheap ones that you can buy off the internet from China. Okay, so I've spoken about bioavailability processes, but it's expensive, there's ethical challenges. We can do something in the laboratory to test the bioaccessibility. This is the amount that is released from the contaminated matrix using simulated gastrointestinal fluid, lung fluid, and so on, to get an idea of what may be absorbed into the bloodstream. Now, if we're talking about the gastrointestinal tract, it is complex. So there's a whole range of different constituents in there, different residence times, different pHs and so on. So trying to replicate this is impossible, absolutely impossible. So what these in vitro assays try to do is to simulate certain components. So you can have a gastric phase with low pH conditions, you can buffer it with various things, you can change the extraction time, the solid to solution ratio. You can then transition that into an intestinal phase by increasing the pH, putting bile salts, pancreatin, changing the extraction time. For environmental contaminants, this is new-ish, so it's been around maybe for the last 10, 15 years. But things like a Hansen dissolution apparatus has been used in pharmaceutical studies, it's been used in nutritional studies since the 50s and the 60s. So it's just been adapted for environmental contaminants. Now, the beauty of running these types of assays is that they're rapid, they're inexpensive, they're reproducible, and if you can find a relationship between bioavailability and bioaccessibility and look at a very strong correlation between the two, you can use these as surrogate methods. You can run more assays, you can get greater confidence in terms of the data that's being generated. They're rapid and you can incorporate them into the risk models. We have done this for multiple examples. This is some of the work, a combination of data for arsenic contaminated soils, lead contaminated soils, huge scatter in terms of the bioaccessibility driven by the mineralogy of the contaminant and driven by the mineralogy of the soil or the dust. We've also developed these methodologies for inhalation exposure. So this is some work done by a former PhD student of mine, Fazana Katsuri, that was looking at inhalation exposure. So if we have our dust and if it's inhaled, then there is a certain amount that potentially could be solubilised once it's in the lung fluid and cross that air-blood barrier. But there's a lot of mechanisms for clearance of this type of material, so mucociliary clearance. So there's the potential that the particles could be cleared out and then end up in the stomach. So it's a dual system of inhalation exposure followed by oral ingestion and exposure. So we had a project with Health Canada looking at potential exposure to dust inside the house for rare earth elements. And the study looked at both households that had people that were smokers and non-smokers. And there were differences in terms of the rare earth elements in there driven by the smoking habits of the individual. But what we found, this is for a data set of 25 in the non-smoking households, was that if the material was inhaled, and in this case this is for cerium, a very small amount of that was solubilised that could potentially cross that air-blood barrier. But if the material was cleared out and went into the gastrointestinal tract, that was the major exposure pathway. That's how you were going to be exposed to these in-house environmental contaminants. We've got assays for organic contaminants. This is for polycyclic aromatic hydrocarbons. These are fused benzene rings that are produced from high temperature processes, commonly associated with gasworks sites. Now, every small town and large town and cities in Australia had gasworks sites historically. They were often in the centre of town. We're not producing energy from that source anymore. So these are prime real estate areas. So people want to know, developers of these areas, is it fit for people to live on the site? Now, again, the standard approach for doing a risk assessment is you will assume that all the contaminant in the soil will be absorbed into the systemic circulation. The thing that we need to worry about is the benzoiopine toxicity equivalence. This drives potential cancer risk. And we found when we assessed over 100 soils that the upper 95% confidence interval was about 10%. So again, you may be overestimating risk by a factor of 10. That may have consequences in terms of your remediation strategy, the cost to redevelop the area and so on. A similar example, we've developed a methodology for dermal absorption. This is where we have a pseudo skin that again looks at the release of the contaminant and potential for it to cross the dermis into the bloodstream. And again, looking at our benzoiopine toxicity equivalence, it is at least an order of 10 less than the standard guideline value used for assessing contaminated sites and risk. Oh, jumped too quickly. Now, as I mentioned before, the value in these assays is looking at the relationship. If there's a very strong relationship between in vivo and in vitro outcomes, then you can utilise the in vitro assay, the simple, rapid, inexpensive one, as a way of having greater detail in your risk assessment. So this is what some work that we did collaborating with the US EPA, a large data set of animal studies looking at arsenic relative bioavailability, compared that to our in vitro assay, a very strong relationship between the two. And now it's in a prescribed methodology by the US EPA for assessment of contaminated sites, and it's also included into guidance in Australia. This has been adopted by both regulators, industry groups, consultants in Australia, and as a way of acknowledgements, a number of these different groups have been funding the research that myself and my team have been undertaking in contaminant bioavailability. So just as a conclusion, why is bioavailability important? We want to understand the internal dose. We want to understand how much can potentially have an impact on an individual prior to the individual being at the contaminated site. Now, if there is retrospective analysis, we can utilise Emily's methodologies for biomonitoring the individual, but this is trying to get an understanding of what is happening before people get on contaminated sites or potentially redevelop contaminated sites. So it's the effective dose. So we can undertake assessment, come up with these bioavailability contaminant maps, understand that there's greater risk in these areas compared to in here, and that's where the remediation effort goes, to clean up that. You spend the money where the effort is required. So I'll leave you with this cartoon. Unfortunately, in the areas of contaminant bioavailability, there are very few cartoons. But this is one by a scientist called Nick Kim in New Zealand. I was fortunate enough to work with him on an arsenic bioavailability project, just to give you another appreciation of contaminant bioavailability. So thank you very much.
We have a few moments for questions before I, on your behalf, thank the speakers. We have one question already.
Thank you. For some time, lead levels in children's blood around Port Pirie have been monitored without much improvement, it seems, over a number of years. What more should they be doing? And should this be applied to places like Broken Hill, where the source of the ore is?
Look, that's a great question. The issue with Port Pirie has been there for 125 years. There is voluntary blood lead monitoring of children, which some families do take that up. But as indicated by the SA Health blood monitoring data, just in the last year, it's jumped up. So there's issues associated with emissions coming out of the smelter. For individuals, there has to be a multi-pronged approach. And the SA Health have started this in terms of over the last 10, 12, 15 years, looking at educational programs for families to really press home the issue about personal hygiene, cleanliness of the house, because the house can be a source of exposure. There also has to be programs put in place in terms of what is coming out of the smelter. And the redevelopment in part is trying to address that with the new smelter facilities. I have a project with the Targeted Lead Abatement Program that is trying to get a better understanding of how the kids are exposed. So we don't know whether the exposure is coming from inside the house, whether it's coming from outside the house, whether it's when they're at school, whether it's a combination of those. So if we can monitor exposure sources in those different locations, we can have an understanding of bioavailability. We can have an understanding of factors that will influence the bioavailability, such as the speciation, particle size, and so on. Then we'll be able to get a greater understanding of what's the pressing issue for that exposure. Is it, the kids playing outside? Is it just the inside environment? Is it lead paint that's in the house as opposed to the smelter emissions? So we need to have a consolidated approach to identify those different sources. Once you know what the sources are and the predominant pathways, then you can start implementing risk management strategies.
Further questions?
Other questions?
You had a question? Any further questions?
If I may. I would like to just invite both speakers to... Oh, one
Could I
more.
just
Yes,
quickly,
certainly.
Just quickly ask, of the 28 PFAS that you know about, what are they examined like in combination? Like, so you know what happens if there's 1? What if there's 4? What if there's 8? What if there's, 28?
That's a fantastic question that I don't think anyone in the world can answer. So thanks for putting me on the spot. Look, it's a complex issue because even if you're looking at, for example, firefighting foams, the predominant compound that's in there is PFOS. So it's the 8 carbon chain length sulfonic acid. Now, it may comprise 85% of the total PFAS concentration, but you have all these other compounds in there. So it could be a 6 carbon chain length sulfonic acid. It could be other things. How they interact and what the effect of that interaction is, we don't know.
That's very heartening.
I think you'll agree with me that we've had two fascinating presentations which demonstrate research that's being carried out that's immediately applicable to the quality of human life, the efficacy of health, diagnostic techniques and the like. I would like to just say, before I ask both Emily and Albert to stand here, that we do have a small gift for them, which I think perhaps illustrates that there can be some positive contaminant effects. We're going to give them a small gift of Haig's chocolates.